DNA replication explains how DNA could be copied or replicated. This video will describe and demonstrate the process. The nucleus is located in the cell, and in the nucleus of each cell, the DNA molecule is packaged into thread-like structures called chromosomes. Each chromosome is made up of DNA that is tightly coiled around proteins called histones that support its structure. The DNA and histone molecule form a structure called a nucleosome. Genes are simply short regions of DNA that code for a certain protein or RNA molecule. The genes on each chromosome have a particular location on the chromosome. Now that we know the basic structure, let's take a look at the actual replication process. Because each strand of the DNA double helix can be used to make the other strand, the strands are said to be complementary. If you could separate the two strands, the rule of base pairing would allow you to reconstruct the base sequence of the other strand. The complementary base pairing rules in DNA are that adenine goes with thymine and cytosine goes with guanine. DNA replication proceeds in both directions until each chromosome is completely copied. The site where separation and replication occur are called replication forks. Before dividing, the cell duplicates its DNA in a process called replication. Each strand acts as a template. The DNA unzips, and enzymes such as DNA helicase open up the helix. Free nucleotides pair up with exposed nitrogen bases on template according to the base pair rules. The DNA polymerase enzyme ensures that complementary base pairing has been done correctly, and these nucleotides are held together by hydrogen bonds. Each new strand of DNA produced contains one old parent strand and one new daughter strand. DNA ligase is an enzyme that functions during DNA replication and DNA repair. It functions by filling in the gaps in DNA that are created when DNA is synthesized. DNA replicates by semi-conservative replication, which means that two strands of the DNA helix separate to form two new DNA molecules known as the parent and daughter strand. At the end, two complete double helical DNA molecules are made, and this marks the end of the process.